So this is the final part of Lab 3, and this is dealing with eukaryotic cells. So we're going to use yeast as our model organism. So we're going to make a yeast wet mount and stain those, and then in the next lab, Lab 4, we will assess the yeast structure. The first thing we're going to have to do is make a yeast solution. So we're going to get some warm water and heat that up to a little over 100 degrees Fahrenheit. And then we're going to add equal amounts of sugar to a yeast. So this is 1 teaspoon of sugar followed by 1 teaspoon of yeast. And this will activate the yeast and get them growing a little bit. And so it needs to sit for 10 to 20 minutes. And to know if the yeast is living or not, you should start to see bubbles and kind of a foamy surface on top. And so that would indicate the yeast is alive. Okay, so the final staining procedure is going to revolve around live yeast cells. So I did this some time ago. I've already activated this, and so if you don't remember how to do that, it's in the procedure, but you take about a half cup of warm water. So you don't want it hot, but you want it warm. You add a teaspoon of sugar. That's going to be the nutrient source for the yeast, and then a teaspoon of the yeast that we provided to you guys. And after about 10 minutes time, you should see some foaming and bubbles form. That would indicate that the yeast is alive and it's actually metabolizing that sucrose. And that's going to be what we're going to make our last slide. And so what I have in front of me, you can see this is my space that I used to do my previous staining. So this is the last activity. It's called a yeast wet mount. And so I have a slide here. I also have a cover slip. So these are the small glass cover slips. I have my loop again, and this is still isopropyl alcohol. The staining solution that you're going to need is methylene blue. So that's what we have. And to apply that, you're going to need another transfer pipette. So because we're going to get a sample of microorganism, we're going to use our loop. So I have my tea light that I'm going to light for this. Okay, so I'm going to sterilize my loop by putting in the isopropyl alcohol for about 10 seconds. And then once that has been done, I'm going to put that into my tea light and get it glowing hot. And then we're going to let that cool. So what we want is basically 1 loopful of our yeast solution. So that has yeast cells suspended within it. And so by just dunking your loop in there, you're going to get some yeast in that solution that you transfer, and then you're going to just plop that onto your slide. Okay, so that should be cool. So I'm just tapping my loop. The loop serves much like, I don't know if you call it a bubble wand that you use to blow bubbles with. So there'll be a solution trapped in the middle of that loop. And so if you touch it to the glass slide, it will transfer. So you don't want the liquid to still be on your loop. You want it onto your slide. So I'm just going to put this back and burn off any residual yeast cells. So get that glowing red hot. And then we're just going to add a drop of methylene blue using our non-sterile. So this is just going to go right onto where the yeast were. Just 1 drop. And then we're going to use my cover slip, if I can find it, because everything is clear. There it is. So this is just going to drop right on the surface, and it's going to create, let's see if I can move that. a light blue surface with the yeast in the middle. So this is called a wet mount because it's still wet under the cover slip, and so those yeast cells are actually suspended in a thin film of water between the glass slide and the glass cover slip. So they're actually floating there. The other slides that you made today, we made the smear and then we allowed them to air dry, so they're not considered wet mounts, but this one would be. So we could put this under the microscope and then visualize these yeast cells. So remember the difference between yeast cells, which are eukaryotic, and the bacteria that we put onto our slides earlier. Eukaryotic cells have several differences that we've been highlighting throughout the course. Eukaryotic cells tend to be larger than prokaryotic cells, so these cells are going to be considerably larger when you see them. Again, you're not going to look at them directly under the microscope, but you're going to get images of this for the next lab. So the cells will be larger. Yeast cells tend to be pretty much round. They're kind of globular in shape, so they can be a little bit oblong, but they're pretty much spherical, elongated spheres. Bacteria can vary in their size, or vary in their shape, excuse me. So we've talked about the shape that bacteria can exhibit. So there are round bacteria, there are rod bacteria, there are spiral shaped bacteria, etc. So at the end of this activity, what you're gonna have are several slides that you can take a picture of to illustrate that you've completed this lab activity. So you have 2 simple stains that we did first. So I have simple 1, simple 2, those are stained with crystal violet. I have my 2 negative stains here. So those have now air dried, so -1, -2. And then I have my 2 gram stains, gram stain 1, gram stain 2. So those are the 6 different bacterial smears and stains that we did. And now we also have a yeast wet mount. So make sure you take a picture of those because that's where this lab ends. So the microscopy, unfortunately, we have to do in a more virtual experience. And so that's what the next lab is going to do. That's lab number 4. So remember, like from all labs, you want to make sure you photograph the end product. So in this case, with all of the staining you've done, you're going to have 7 slides to take a photo of. And then once you've done that, we can discard the slides, so they'll be wrapped in parafilm and discarded. The only solution we need to save from the staining is the iodine. So make sure you don't discard the iodine. We need that for a future lab, but all the other stains we do not need. And then, of course, because we're done with the lab, you want to make sure you disinfect your surface that you are working on, and then also wash your hands.